In the quiet corridors of a modern hospital, danger does not always announce itself loudly. Sometimes it creeps silently along the very devices designed to save lives. The IV lines, the ventilator tubes, the catheters. Among the most cunning of these invisible threats is a bacterium that has mastered the art of survival in environments where most microbes would perish. Its name is Pseudomonas aeruginosa, and it glows with a characteristic green light, a pigment it proudly produces as a signature of its presence. This is the story of an opportunistic predator that strikes not the strong, but the vulnerable. Welcome to Pseudomonas aeruginosa, the opportunistic hospital raider. Every name in science carries meaning, and Pseudomonas aeruginosa is no exception. The genus name Pseudomonas is derived from two Greek words, pseudo, meaning false, and monas, meaning a single unit or entity. Together, they suggest something that appears to be a simple unit, but is, in reality, far more complex and deceptive than it seems. The species name aeruginosa comes from the Latin word for verdigris, the distinctive blue-green color of oxidized copper, a direct reference to the striking pigments this organism produces. Taxonomically, Pseudomonas aeruginosa is classified as a gram-negative bacillus, meaning it has a thin peptidoglycan cell wall surrounded by an outer membrane. It belongs to the family Pseudomonidaceae and is the most clinically significant member of its genus, notorious worldwide for causing difficult-to-treat hospital-acquired infections. Under the microscope, Pseudomonas aeruginosa reveals a form that is elegantly simple, yet functionally powerful. It appears as a slender, straight, or slightly curved rod, measuring approximately 1 to 3 micrometers in length and half a micrometer in width. Being gram-negative, it retains the pink counterstain in the gram staining procedure, reflecting the presence of a thin peptidoglycan layer sandwiched between two lipid membranes. One of its most defining structural features is its single polar flagellum, a whip-like appendage attached at one end of the cell, which confers remarkable motility. This motility is not merely a passive trait. It is an active virulence mechanism that allows the bacterium to swim toward host tissues, navigate biofilm environments, and colonize new surfaces with speed and precision. Pseudomonas aeruginosa does not form spores, and it carries no capsule in its free-living state, though it can produce a polysaccharide capsule-like structure under certain conditions, as we will see when we discuss biofilm formation. Pseudomonas aeruginosa is a metabolic generalist of remarkable versatility. It is an obligate aerobe, meaning it requires oxygen for growth, yet it can survive in remarkably low nutrient environments that would starve most other bacteria. It grows optimally at 37 degrees Celsius, the temperature of the human body, but it can also grow at temperatures as high as 42 degrees, a trait that helps distinguish it from closely related species. One of its most visually striking characteristics is its pigment production. The blue-green pigment known as pyocyanin, derived from the Greek words for blue and green, is a phenizing compound unique to Pseudomonas aeruginosa and is a key diagnostic marker. A second pigment, pyoverdine, imparts a yellow-green fluorescence under ultraviolet light. These pigments are not merely cosmetic. Pyocyanin actively contributes to virulence by generating reactive oxygen species that damage host tissues. Colonies of this organism also emit a distinctive sweet grape-like odor caused by the volatile compound 2-aminoacetophenone, a clue that experienced microbiologists can detect even before biochemical testing is complete. Pseudomonas aeruginosa is one of the most environmentally ubiquitous bacteria known to science. It thrives in soil, fresh water, and marine environments, and it has been isolated from plant surfaces, food products, and even the surfaces of medical equipment. Within the hospital setting, it colonizes moist environments with particular tenacity. Hospital sinks, water taps, humidifiers, respiratory therapy equipment, and the water reservoirs of ventilators are all well-documented reservoirs. It can survive in disinfectant solutions, antiseptic preparations, and even distilled water, a testament to its extraordinary metabolic adaptability. Transmission in healthcare settings occurs primarily through contact with contaminated water sources, the hands of healthcare workers, and contaminated medical devices. Pseudomonas aeruginosa is among the leading causes of nosocomial or hospital-acquired infections worldwide, accounting for a significant proportion of ventilator-associated pneumonias, surgical site infections, and bloodstream infections in intensive care units. Pseudomonas aeruginosa is, at its core, an opportunist. In healthy individuals with intact immune defenses, it rarely causes serious disease. But when those defenses are compromised, whether by illness, injury, or medical intervention, this organism seizes the opportunity with devastating efficiency. 4 groups of patients face the highest risk. First, burn patients whose loss of skin integrity removes the body's primary physical barrier, allowing Pseudomonas to colonize wound surfaces and invade the bloodstream. Second, patients with cystic fibrosis, in whom a genetic defect in chloride transport leads to the accumulation of thick, viscous mucus in the airways, a perfect medium for chronic Pseudomonas colonization that progressively destroys lung function over years. Third, patients with neutropenia, a dangerously low neutrophil count, often resulting from chemotherapy, who lack the cellular soldiers needed to contain bacterial spread. And fourth, critically ill patients in intensive care units, particularly those on mechanical ventilation, whose bypassed upper airway defenses and prolonged hospitalization create a perfect storm for infection. The clinical danger of Pseudomonas aeruginosa lies not in a single weapon, but in an entire arsenal of virulence factors that work in concert to establish infection, evade immunity, and destroy tissue. Chief among these is exotoxin A, a protein toxin that operates by the same molecular mechanism as the diphtheria toxin produced by Corynebacterium diphtheriae. It enters host cells and catalyzes the ADP ribosylation of elongation factor 2, a protein essential for ribosomal protein synthesis, bringing cellular protein production to a halt and triggering cell death. Elastase, encoded by the LASB gene, degrades elastin, collagen, and immunoglobulins, breaking down the structural scaffolding of tissues and dismantling key components of the immune response. Phospholipase C disrupts cell membranes by cleaving phospholipids, releasing nutrients for bacterial growth while damaging host cells. And perhaps most importantly for chronic infections, Pseudomonas aeruginosa produces alginate, a viscous polysaccharide that forms the structural backbone of its biofilm. Together, these factors transform a simple environmental bacterium into a formidable pathogen. Of all the survival strategies employed by Pseudomonas aeruginosa, none is more clinically consequential than its ability to form biofilms. A biofilm is a structured community of bacteria encased within a self-produced matrix of extracellular polymeric substances, primarily alginate in the case of Pseudomonas, that adheres tenaciously to surfaces. On medical devices such as urinary catheters, central venous lines, and endotracheal tubes, biofilm formation transforms the device into a protected bacterial fortress. Within this matrix, bacteria communicate through a process called quorum sensing, releasing and detecting chemical signals that coordinate gene expression across the entire community. Bacteria within a biofilm exhibit dramatically altered physiology compared to their free-floating counterparts. They grow more slowly, express different genes, and become up to 1,000 times more resistant to antibiotics. The biofilm matrix physically impedes antibiotic penetration, while the slow metabolic state of embedded bacteria renders many antibiotic mechanisms ineffective. This is why Pseudomonas infections associated with indwelling devices are so extraordinarily difficult to eradicate, and why device removal is often a necessary component of treatment. The clinical manifestations of Pseudomonas aeruginosa infection span virtually every organ system, reflecting both its environmental ubiquity and its capacity to exploit any breach in host defenses. In the lungs, it is the leading cause of ventilator-associated pneumonia in intensive care units, producing a necrotizing infection characterized by fever, purulent secretions, and progressive respiratory failure. In burn patients, it colonizes wound surfaces and produces the characteristic blue-green discoloration of pus, a direct result of pyocyanin secretion, before invading the bloodstream to cause life-threatening bacteremia. In the urinary tract, it is a frequent cause of catheter-associated infections, particularly in patients with prolonged catheterization. In the ear, it causes otitis externa, commonly known as swimmer's ear, an infection of the external auditory canal that is particularly prevalent in swimmers and diabetic patients. In the eye, it causes keratitis, a rapidly progressive and potentially blinding corneal infection, most often associated with contact lens wear. And in immunocompromised patients, it can cause eczema gangrenosum, a distinctive skin lesion characterized by a central black necrotic eschar surrounded by erythema, which is considered pathognomonic for Pseudomonas bacteremia. The laboratory diagnosis of Pseudomonas aeruginosa relies on a combination of culture, morphology, and biochemical characterization that together produce a highly distinctive profile. On blood agar, colonies are large, flat, and spreading, often producing a zone of beta hemolysis, the complete lysis of surrounding red blood cells, reflecting the organism's production of hemolysins. On McConkey agar, Pseudomonas aeruginosa grows as a non-lactose fermenter, appearing as pale or colorless colonies against the pink background of the medium, a critical distinction from the Enterobacteriaceae, which typically ferment lactose. The most selective medium for its isolation is citramide agar, which contains the detergent citramide, a compound that inhibits most other bacteria while allowing Pseudomonas to grow and fluoresce, brilliantly green under ultraviolet light. Biochemically, the organism is oxidase positive, a key differentiating feature from the Enterobacteriaceae, and it is a non-fermenter of glucose, relying instead on oxidative metabolism. The combination of green pigment production, oxidase positivity, characteristic grape-like odor, and growth on citramide agar makes the identification of Pseudomonas aeruginosa one of the more visually satisfying exercises in clinical microbiology. Pseudomonas aeruginosa occupies a uniquely dangerous position in the landscape of antimicrobial resistance. It possesses an extraordinary array of both intrinsic and acquired resistance mechanisms that render it resistant to many of the antibiotics routinely used against other gram-negative bacteria. Its intrinsic resistance begins with its outer membrane, which has unusually low permeability, restricting the entry of hydrophilic antibiotics that would otherwise diffuse freely into susceptible organisms. Compounding this is an extensive network of efflux pumps, particularly the MEX-AB OPRAM, MEX-CD OPRJ, and MEX-XY systems, which actively expel antibiotics from the bacterial cell before they can reach their intracellular targets. The organism also produces chromosomally encoded AMP-C beta-lactamase, which can be stably repressed under antibiotic pressure to confer resistance to broad-spectrum penicillins and cephalosporins. Beyond these intrinsic mechanisms, Pseudomonas aeruginosa readily acquires additional resistance genes through horizontal gene transfer, including metallo-beta-lactamases that inactivate carbapenems. The result is a pathogen that the World Health Organization has designated as a critical priority organism for the development of new antibiotics, a reflection of the genuine therapeutic crisis it represents. The treatment of Pseudomonas aeruginosa infections demands a strategic and often aggressive approach, guided at every step by antibiotic susceptibility testing. Given the organism's formidable resistance profile, empirical therapy must be carefully chosen from the limited arsenal of anti-pseudomonal agents. Beta-lactam antibiotics with anti-pseudomonal activity, including piperacillin tazobactam, ceftazidime, and cefepime, are frequently used as first-line agents, targeting the penicillin-binding proteins involved in cell wall synthesis. Carbapenems such as imipenem, meropenem, and doripenem offer broader coverage and are often reserved for more severe infections or resistant strains. Aminoglycosides, including tobramycin, gentamicin, and amikacin, act by disrupting ribosomal function and are frequently used in combination with beta-lactams, both to broaden coverage and to reduce the emergence of resistance. Fluoroquinolones, particularly ciprofloxacin, remain one of the few oral agents with reliable anti-pseudomonal activity. Crucially, combination therapy is often preferred for serious infections, not only to achieve synergistic bacterial killing, but to prevent the rapid emergence of resistance that can occur when Pseudomonas is exposed to a single agent. All treatment decisions must ultimately be guided by the individual patient's culture and sensitivity results. In the battle against Pseudomonas aeruginosa, prevention is not merely preferable to treatment, it is essential. The organism's environmental resilience, its capacity for biofilm formation, and its resistance to many disinfectants mean that once it establishes itself within a hospital environment, eradication is extraordinarily difficult. The foundation of prevention lies in rigorous hand hygiene. The single most effective measure for interrupting the transmission of healthcare-associated pathogens, alcohol-based hand rubs and soap and water hand washing, performed consistently at every point of patient contact, dramatically reduce the risk of cross-transmission. Equally important is the meticulous care of invasive medical devices. Catheters, ventilator circuits, and foreign lines must be inserted under sterile conditions, maintained according to evidence-based protocols, and removed at the earliest clinically appropriate opportunity. Environmental surveillance, including regular microbiological sampling of water sources, sinks, and respiratory equipment, helps identify reservoirs before they become sources of patient infection. Contact precautions, including gloves and gowns, are essential when caring for patients colonized or infected with multi-drug-resistant strains. And antimicrobial stewardship, the disciplined, evidence-guided use of antibiotics, remains our most powerful tool for preserving the efficacy of the agents we still have. Against Pseudomonas aeruginosa, vigilance is not optional, it is our best defense.